So, in our last discussion, we said that aging is one of the most well studied phenomena in biology, and there are more than 300 theories of why aging happens. We also said that it is not possible for us to look at all those theories, and we will look at just a few more prominent ones. So, if we look at the theories of aging, there are different classes of them, and there are two broad classes, the so called mechanistic theories of aging and the so called evolutionary theories of aging. So, in this discussion we are going to look at the first category of theories, the so called mechanistic theories of aging, and as I said, we are only going to look at a few more prominent ones. So, one of the well known mechanistic theories is the so called rate of living hypothesis, which was proposed by Raymond Pearl in 1928. So, this is the same Raymond Pearl who gave the logistic model of population growth. So, he has made very significant contributions in the field of population dynamics as well, but in this particular case we are more interested in what he had to say about the aging and why aging happens. So, what Raymond Pearl said was very simple. He said that look, aging is primarily determined by the pace of living, in other words the metabolic rate, right. And he said that species that have higher metabolic rate, they age or die faster. Okay. So, think of it as candles, let us say all the candles have the same height. So, they have the same amount of wax in them, but some of them are burning faster and therefore they get over faster. Some of them are burning at a slower rate and therefore they last longer, right. So, it is pretty much exactly the same thing with organisms. Those which are spending their energy at a higher rate and therefore have a higher metabolic rate, they are the ones who are expected to age or die faster. Now, how are you going to check this out? Fortunately, checking this out is very simple. There are all kinds of ways in which one can measure the metabolic rates of organisms. So, all you have to do is to take a bunch of organisms and measure their metabolic rates, measure their average lifespan and see if there is a correlation between that or not. And this is exactly what was done by Orsted and Fisher in 1991. So, what they did was they looked at the amount of energy spent per gram tissue per lifetime, and they did this for 164 mammalian species spread across 14 orders. And what they found was, or rather what they predicted was, that mammals which have a higher metabolic rate, they will have a lower lifespan and vice versa. Easy prediction. But what they found was that bats, remember these are flying mammals and therefore they have the highest metabolic rates, no surprise over there, but they also tended to live 3 times longer than the other mammals. So, obviously, this is not squaring with the prediction. Similarly, they saw that marsupials, the kangaroos and other pouched animals, they have the lowest metabolic rates, yet they also have the lowest longevity, and neither of these two observations support the rate of living theory. Now, of course, one can say that this is just about mammals, maybe it is not true in mammals, maybe it is true in other kinds of organisms. So, people then looked across the life table of different organisms, and what they roughly found looked like this. So, if you take birds and mammals, remember birds are mostly flying and therefore they have a much higher metabolic rate, and therefore you expect them to have a lower longevity, but in reality it is the birds which have the higher longevity compared to the mammals of corresponding sizes. So, not working out. Similarly, if you look at ectotherms, so you remember that there are 2 kinds of organisms, ectotherms which cannot maintain their own body temperature through endogenous mechanisms, and endotherms like for example mammals and birds who can maintain their body temperature through internal mechanisms. So, ectotherms in general have a lower metabolic rate, and therefore one would expect them to live longer if the rate of living theory were correct, but in reality the ectotherms also have a lower longevity, which basically means that this is not working out in the context of rate of living theory. And then finally, in 2007 there was an analysis of again mammals and birds. This is 467 species overall, and they ended up rather conclusively showing that there was no correlation between basal metabolic rate and longevity. So, taken together, now it is reasonably clear that the rate of living theory is not supported by any kind of observations. Okay. So, what about other kinds of mechanistic theories? So, one other class of mechanistic theories are the so called wear and tear hypothesis, and this basically says that aging is due to some kind of accumulation of damages to cells and tissues. So, this is coming directly from thinking of organisms like machines, like a car for example. So, the whole idea is that if you use a machine for a long time, then it is going to accumulate small tears, small imperfections or small wears and so on, and over time all these are going to accumulate and make the machine non-functional like a broken down car. And that is exactly what is happening to organisms. Due to the functioning of organisms, there are all kinds of damages to cells and tissues. For example, you can have damages in terms of toxic metabolic byproducts, for example reactive oxygen species. So, this was a theory that was given in 1956 by a guy called Harman. Or you can even have wear and tear in terms of DNA damage. So, this was a theory which was propounded by this very famous physicist called Leo Szilard. So, this was a Hungarian physicist and he is extremely famous because he made fundamental contributions to nuclear physics. More importantly, he was the person who drafted the letter which was then signed by Einstein and sent to the American President Roosevelt, which finally leads to the Manhattan Project, finally leading to the atom bomb. So, basically Leo Szilard was the person who was very directly responsible for reducing the longevity of lots of human beings on this planet. Not his fault, I obviously do not want to blame him over here, but he also ended up making very fundamental contributions in multiple other subjects including biology. And this was one of his contributions to biology. He gave this theory that there are cumulative damages that happen to the DNA over time, because of which organisms age. So, we are going to look at both these theories in some amount of detail. So, let us first look at the free radical theory. So, what is it saying? So, it is saying that due to cellular respiration, there are these things known as reactive oxygen species, also abbreviated as ROS, H2O2 for example. These are the molecules that damage all sorts of cellular components. Now, these are typically produced, as I said, in the mitochondria as a normal byproduct of cellular metabolism. Now, the thing is that all cells have multiple ways of taking care of these molecules, because if they cannot take care of these molecules, they are essentially going to die. So, for example, all cells have antioxidants like vitamin C and vitamin E, molecules which directly quench these ROSes. You also have enzymes like catalyses and superoxide dismutases which protect against these ROS. So, it has been shown that if you look at the oxidative damage in various tissues, then the extent of that damage increases as the age of the tissue increases. Okay. So, this is one of the major reasons for which people thought that this is what is leading to the aging. Now, look at how things happen. What has been seen is that as the tissues age, there is more oxidative damage. The free radical theory is inverting this and is saying that because there is more oxidative damage, hence there is aging. Okay. Now, the question is, does it work? Can we do this inversion, logical inversion? Now, if in order to test this theory, if the aging is indeed due to reactive oxygen species. Remember, aging is the deterioration of the function, right. So, if the aging is indeed due to ROS, then that basically means that whatever antioxidant enzymes or antioxidant molecules like vitamin C and vitamin E we have, that is not being sufficient, correct. So, if that be the case, that means that if you increase the amount of these enzymes or if you increase the amounts of vitamin C and vitamin E, then that should reduce aging or increase lifespan or maybe both, right. If the amount of the chemical is limiting, then you increase the amount of the chemical and then that should prevent aging. Alternatively, if you take the existing amount of these chemicals and somehow reduce them, okay, then the other prediction is that that itself should increase aging or lead to a reduction in lifespan. Okay. So, very often this aging and lifespan, they are kind of taken studied simultaneously, synonymously with each other. Now, how will you test this? Again, testing is relatively easy. So, for example, you know that lots of people, or it is very easy to give these kinds of antioxidants like vitamin C and vitamin E through the diet. And this is something that has been done for a very long time, these experiments, and turns out that there is either weak or zero evidence to indicate that dietary supplementation of antioxidant molecules influence the aging process. Okay. So, here is a review for that, you can look it up if you are interested. Okay. Then there were lots of experiments that were done using genetic manipulation. So, in one such experiment, what Remen et al. Did was they looked at mice in which there is only one functional copy of the superoxide dismutase gene. Remember, superoxide dismutase plays a role in cutting down the ROS molecules, getting rid of the loss molecules. So, if that be the case, then you expect such mice to show greater amount of oxidative damage. Turns out they see that. But now if you look at the aging in these animals or the lifespan in these animals, it turns out that neither of them are significantly different from the controls, which basically means that the oxidative damage is happening, but the extra oxidative damage is not really leading to a change in the lifespan or aging, which basically says that these two are not causally related. Oxidative damage is not causally related to aging. And there are many more such studies. So, here is a quick small review, well, it is not a review, it is actually a chapter on aging, but it has a very nice section on the free radical theory and it gives you more examples. Basically what happens is that although it has become clear that although ROS can play a role in the aging related pathologies, on the aging related negative effects that we see, there is little reason to believe that it is causing the aging to begin with. Okay. In other words, that logical inversion that we were talking about, that aging is because of ROS, there is no evidence to suggest that. Okay. So, suppose it is not ROS, we also had one more, which was DNA damage. Okay. So, it has already been seen that if you look at older animals, then at least for some of the tissues that these animals have, they seem to contain more amount of mutations and more amount of DNA lesions in them, okay, breaks in them at a higher frequency. Now, this obviously means that DNA damage is something that is known to happen. But if that be the case, does that lead to aging? Can we connect that to age, or can you say that it is a cause of aging? So, it turns out that if you take mice which overexpress proteins that are related to DNA repair, okay, remember the body has all kinds of repair mechanisms which take care of whatever mutations happen during DNA replication. So, if you overexpress proteins that will lead to this DNA repair, you expect that there is more repair happening, therefore less aging. Yet such animals, such mice, they have the same lifespan, they do not have an increased lifespan and their aging process is also similar. Okay, so the pathologies also do not show up any differently, suggesting that probably there is no causal relationship. Now, this particular theory of DNA damage, it has not been conclusively proven or disproven, direct damage to DNA, and the evidences are equivocal. However, there is a class of, there are many kinds of DNA damages. Okay. So, one of the major kinds of DNA damages that can happen and has been implicated in the context of aging is the so called telomere shortening. Okay. What is the telomere? So, telomeres are basically regions of repetitive nucleotide sequences which are located at the end of a linear chromosomes in almost all eukaryotes. Okay. So, just to show you a picture. So, you can see the chromosomes over here, and can you see these white dots at the ends of the chromosomes which have been pointed with the two white arrows? These are the telomeres. Okay. Now, what is the function of a telomere? So, turns out that they actually prevent the DNA repair systems from misinterpreting the ends of the DNA strands. Okay. So, what happens is, as I said, we have lots of repair mechanisms for mutations in our body. So, many of these repair mechanisms, whenever they see a break in a DNA, they assume that there is a break and they end up repairing that. Now, if you come to the end of the DNA, obviously it is a break, right. So, the DNA repair mechanism might end up interpreting it as a break and might start trying to repair it, which is a problem. So, the telomeres prevent that, and by preventing that, they act like protective caps and they safeguard the terminal regions of the chromosomal DNA from degradation, because otherwise these DNA repair mechanisms, they can actually end up marking it for destruction, that particular DNA for destruction. The telomeres prevent that. Now, it is known that at every replication a small part of the telomere is lost. Okay, you can't do anything about it. Now, when the length of the telomere becomes too short, there are certain enzymes known as sensor kinase enzymes. They activate a particular protein called a transcription factor called p53. Now, when this particular protein gets activated, there are many other cascading reactions that happen, other proteins get activated, and because of the whole thing, the cell can become non-dividing, or it can even undergo programmed cell death known as apoptosis. Now, you can imagine that this is one of the mechanisms by which the body actually fights cancer, right, because cancer is what? Uncontrolled growth of cells. So, p53 is able to take certain cells and then they can mark them for destruction, okay, but or arrest their growth. But the arresting of growth or the death of the cells is also something that is related to aging, right. So, the hypothesis here is that organisms age because over time their telomeres become too short, and when that happens, the chromosomes and hence the cells, they become too damaged to function normally. So, they basically become non-functional or misfunctional, and at that point the organism starts showing all kinds of symptoms of aging. Okay. So, if that be the case, then what is my prediction? My prediction is that if I have longer telomeres, then it will take it much longer to degrade that telomere to a level at which p53 will start acting on it. Therefore, longer telomeres should imply greater longevity, right. So, a positive association between telomere length and longevity. Do I see that? So, turns out that in a few cases it has indeed been seen. Okay. So, for example, here is a bird, zebra finches, very well known bird, and these it has been shown that the finches which have a longer telomere length at 25 days of age, they seem to have a much greater lifespan. Okay, and it is a statistically significant relationship. Okay. So, this suggests that there might be some truth to it, but then is this something that has been replicated? Is this in other systems? Have we seen it in multiple cases? And turns out that this is where things become complicated. So, for example, there was a study of telomere length across 57 mammalian species, and what they found was this. So, on x axis you have the length of the telomere, on y axis you have the maximum lifespan. You expect a positive relationship. What you are seeing is a very clear negative relationship, right, suggesting that the longer lived mammals are the ones who actually have the shorter telomeres. It is just a reversed, right. So, then people sought to look at it in other kinds of organisms, and again the story became very, very complex. So, for example, if you look at multiple strains of this worm called Caenorhabditis elegans, then it is a very famous model system and has been used extensively in the context of aging research. In those, you actually do not find a relationship between telomere length and longevity across strains. In a few strains you might end up finding it, but if you look at many strains you are not going to find it. Similarly, if you look at the telomere length and longevity relationship between in humans, you are actually going to see that there is no such correlation. And this is very interesting. This is a study on mice, and what they did was they took wild derived strains, and these are strains had shorter telomeres, and yet if you look at their longevity, the longevity was not significantly different from laboratory strains which had obviously a longer telomeres. So, taken together, it is now more or less established that there is no relationship between aging and telomere shortening. I mean, there are a few studies which do report some relationship, but there is no general relationship across the board. Now, please appreciate one thing. I have already said that telomere shortening is one of the hallmarks of aging. That is correct, okay. But what I am now saying is that telomere shortening is not a predictor of aging, okay. Or shorter telomeres do not mean that aging is going to be accelerated, okay. Both the statements are correct. As an organism ages, its telomere gets shortened, but if you have short telomere to begin with, that does not mean that you are going to live less, okay. That is the point that needs to be taken from here. So, we do not seem to have any good consistent evidence for any candidate for the wear and tear theory, but there is a problem. What is the problem? The problem is that if you do not have proof for something, that is not a proof that that thing does not exist, right. So, what happens if we have not, I mean, the wear and tear theory is correct, it is just that we have not hit upon the correct candidate, or alternatively, what happens if there is a limit to which the errors can be corrected and the organisms have already reached that limit. I mean, if that be the case, then that might explain why when we are supplementing the diets with antioxidants, or when we are increasing the amounts of the enzymes, superoxide dismutase or whatever, why those things are failing to have an effect. So, if we take this possibility, then what is the prediction? The prediction is that all organisms have already reached the limits of their error correction, they have reached the limits of their lifespan, and if that be the case, there is no variation left, genetic variation left, which means that if you now try to select for increased lifespan, then that selection should fail, okay. In other words, what we are trying to say is that the mechanism is correct, it is just that the mechanism is wear and tear mechanisms are the ones which are leading to aging, but they are operating at their limits, which is why if you change the levels there is no effect happening, and the corollary of that is therefore it should not be possible to select for increased lifespan in any organism. So, this is something that of course can be very easily checked for, and it has been checked for in multiple organisms, and particularly good amount of work has been done on the common fruit fly Drosophila melanogaster. So, this is work done by Luckinbill et al. In 1984. So, what they did was they took two replicate populations of Drosophila melanogaster and maintained them in milk bottles, and every 48 hours, the milk bottles had Drosophila food, the fly were on the food, and every 48 hours they would transfer the flies to a different bottle containing Drosophila food, fresh food basically, and they kept on doing this every 48 hours, and every few days, every few generations, they would push back the day on which they are collecting the eggs for creating the next generation, right. So, this is the discrete generation system. So, obviously, after a few days they would collect eggs for the next generation, but the day on which they are doing that collection, they are pushing that back every few generations. Now, what is happening because of that? Remember, in a system like this, reproduction is conditional upon living to a later age. In other words, suppose let us say on day 20 they decide to collect the eggs to make the next generation. So, they are only taking eggs which are laid on day 20, okay, or day 30, whatever. So, obviously, only those flies will be able to lay eggs on day 30 which are surviving till day 30, right. So, that is what I mean by saying that reproduction is conditional upon living to a later age, right. And every few generations they would push back the day on which they are collecting the eggs, from 30 to 35 and 40 and so on and so forth. And they did this for a few generations, and every few generations they would also measure what was the maximum lifespan of the flies, and what they found was very stark. So, when the whole experiment started, the mean longevity of the flies across the two replicates was somewhere about 25 days, and within about 13 generations of selection this had increased to about close to about 50 days or so. So, roughly speaking, within a span of about 13 generations they had doubled the mean lifespan, roughly doubled the mean lifespan. Now, you can imagine that that is a huge change in a very short amount of time. Were they the only people to see such a result? Absolutely not. There were many such experiments. One very famous experiment was done by Michael Rose and people around him, again published in the year 1984, and they ended up showing the same thing, that the longevity of the organisms can be increased via selection. Okay. So, this proved that the limit to lifespan has not really been reached in many organisms, and after the 80s, these kinds of experiments have been repeated in really a large case of organisms, large number of organisms across taxa, and in many of those cases they have been able to replicate this result. So, it is a very strong result, which means that selection can increase the lifespan and the limit has not yet been reached, and therefore, we can conclude that the wear and tear theory definitely is not something that can be generalized. In most of the cases it does not seem to hold. Now, as I said, people at this stage can say that look, we do not know what really is happening, but maybe somewhere it is all in the genes. After all, everything that organisms do we think is in the genes, it is because of their genes, and therefore, it has to be in the genes. And that is one reason for those 3 lakh plus studies, people have been searching madly for genes that will enhance longevity. And why they are doing so? Because, as I said, it is a very, there is a commercial and a social angle to it. So, I can't really blame them entirely, but when it comes to the science, it's it's not entirely clear whether this is leading to something or not, but one thing is clear, that it is leading to a lot of discoveries. So, thousands of genes have been implicated in aging. Implicated means that people have shown that if you knock them out, or if you create a mutation, then that can cause some change in terms of the aging pattern. So, just to give you a comprehensive listing, I have given you this link. As of December 2024, this is the last time that they updated this particular database. There were 2205 genes on this database. Okay. In some system or the other, in some strain or the other, these are the genes which have been shown to have an effect, and yet there is no single gene or set of genes which are implicated for aging across all taxa. However, given the commercial incentives etc., the search is still on. So, this is where we will conclude our mechanistic set of mechanistic theories of aging and our overview of those. And when we come back for our next discussion, we are going to talk about the second class of theories of aging, the so called evolutionary theories of aging. See you then. Bye.